Hello, good afternoon. So we are start our session of the prostaglandins. So first, our first speaker, Professor Shuh Narumiya. Professor Narumiya is a professor of drug discovery medicine and the director of Medical Innovation Center at Kyoto University. Before that, he was a professor in pharmacology of Kyoto University Graduate School of Medicine for more than 20 years, since 1992. He had cloned the all prostaglandin receptor family and separately discovered the botulinum C3 enzyme and discovered the very popular ROCK inhibitor Y27632, and his research has spanned from pharmacology, cell biology, biochemistry, and molecular biology to pathophysiology of disease in the prostaglandins and Rho proteins areas. He has served as the dean of the Kyoto University Graduate School of Medicine in 2004 to 2007, and he had received lots of honors and awards worldwide, and he was the president of last World Conference of Pharmacology, WCP 2018. Now we welcome Professor Shuh Narumiya.
Thank you for your kind introduction. First of all, I thank Xavier and Chengcan for a kind invitation. Good afternoon. Today, I'm talking about recent and unpublished work on roles of PGE2, EP2, EP4 signaling in immune landscape of tumor microenvironment, comparative HC-seq analysis. This is my disclosure of conflict of interest. Right. So prostaglandins, including PGD2, PGE2, PGF-alpha, PGI2, and thromboxanes, are the oxygenated metabolites of arachidonic acid. Because aspirin, like non-steroidal anti-inflammatory drugs, inhibit PG synthesis by inhibiting cyclooxygenases, prostaglandins are believed to function in aspirin-sensitive processes such as inflammation, pain cessation, and fever generation. It is known for some time that the cyclooxygenase, particularly inducible isoform COX-2, is expressed in the tumor tissues, and the expression of COX-2 is associated with poor prognosis of cancer patients. It is also well known that daily use of COX inhibitors, aspirin-like drugs, lowers the risk of cancer death of a variety of cancers. And this prevention is not just prophylactically, but also aspirin-like drugs inhibit cancer death therapeutically. And curiously, that the aspirin effects is more evident in low PD-L1 expression colorectal cancers, indicating there is some interactions between the acquired immunity against cancer and aspirin-sensitive processes. So these findings indicate high potential of COX inhibitors in cancer therapeutics, but drug developments on this line has been hindered by the GI toxicity of general COX inhibitors and the cardiovascular toxicity of COX-2 selective inhibitors. effort has been made on manipulation of PG receptors as an alternative measure. Indeed, EP4 antagonists and EP2, EP4 dual antagonists are now in clinical trials. However, how they act in tumor, particularly tumor microenvironment in vivo, remains largely unknown. We have addressed this issue by carrying out single-cell RNA-seq of immune cells in tumor microenvironment and compare the findings obtained in human samples with that of the mouse model of cancer. Okay, this shows the protocol of our work. We obtained a surgical specimen from 5 patients of colorectal cancer, 5 patients of breast cancer, 5 patients of ovarian cancer, and dissociated cells and the collected CD45 immune cells and subjected them to single-cell RNA-seq. By doing that, we identified cell clusters expressing EP2 and EP4 and their gene expression signatures. In parallel, we use mouse tumor models such as LLC1 tumors or CT26 tumor, and we here intervene the actions of PGE2 by administering EP2 inhibitor and EP4 inhibitor in combination. And again, we collected CD45 immune cells in this tumor microenvironment of mouse tumors and subjected it to single-cell RNA-seq. By doing this, we identified cell clusters altered by EP2 inhibitor and EP4 inhibitor and their gene expression signatures in mouse tumors. And by comparing these two, we examine the causative relationship of human data by mouse model findings. Practically, we collected dissected tumors and dissociated to single cells and FACS-sorted CD4 positive cells, and we subjected it to the 10x, the single-cell RNA-seq protocols, and the single cell and UMR barcoding in the droplets. Okay, then we prepared the cDNA library of individual cells, the sequencing, and we have the LEDs and they perform quality control and alignments and quantification per genes. Then we make use of those data to unbiased clustering of cells and each cluster annotated by expression profiles. Okay. So this is the clustering of the immune cells we collected from 15 cancer patients. That consists of 86,000 cells. And that's we divided into the 9 clusters: NK cells, CD8 T cells, CD4 T cells, regulatory T cells, PMN, TAM, and MMDC, monocytic cells, DCs, B cells, and plasma cells. Okay, we first mapped expression of COX-1 and COX-2 over these clusters and found out that COX-1 and the COX-2s are mainly produced or expressed in the myeloid cells containing TAM monocytes and the TAM in case of COX-2. And what's about EP1 to EP4 expression? We have found expression of EP1 and EP3 were little seen in these clusters, and the EP4 is dominant subtype of the receptors they express by many clusters, including myeloid cells, but also the T cells. EP2 is to less extent are expressed, but also expressed in the T cells and also myeloid cells. But EP2 and EP4 are not expressed by all the cells in the each clusters. Maybe at most about the 50 to 60%. That's indicated that expression of these receptors occurs cell context dependent manner. Okay. We further subclassified the CD8 T cells to the 9 subsets. And we found that among those cells, extent of the PTGR4, EP4 expression is different and could be divided to 4 groups: high EP4 expression, intermediate low EP2 expression, low EP4 expression, and undetected expression. Right? So there are the very inhomogeneous population of cells in terms of EP4 expressions. Because those 4 groups are equally distributed in the 9 subsets, we subjected to the DEG analysis, differential expression gene analysis, as a total of CD8 T cells. Okay. We have found that about 4,500 genes are upregulated in this comparison, and 5,700 genes are downregulated in this comparison. Yeah. So this, we carried out differential expressed gene in high for this low GTPR EP4 expressing CD8 T cells. Okay? Yeah. Because the bar graph that shows that the frequency of the upregulation, for example, in this case, score 15, the bar shows the 15 of 15 patients shows the expression of that genes, right? By comparing these expression, the genes, we extracted the EP4 expression associated genes, okay? And when we subjected these gene signature to GSEA analysis, we have found that the trends is the IL-2, downregulation of IL-2 STAT5 signaling, downregulation of oxidative phosphorylation, and downregulation of MYC target genes. Okay. Here we show the upregulation and downregulation of several clusters of genes with EP4. So EP4 expression is associated with several activation markers of the T cells, such as CD44 and CD69, and also it is associated upregulation of AP1 genes, such as FOS and JUN, indicating that EP4 is expressed in the activated cells. But curiously, EP4 expression is associated with downregulation of TCR IL-2 signaling and also the downregulation of various oxphosphor genes and lysosomal proteins. Okay, what's extent is this association, particularly downregulation of oxphosphor and ribosomes. Okay, so oxphosphor genes encode the molecules molecular components of the electron transport chains, complex 1, 2, 3, 4, 5. And when we examined the expressions of those components in terms of the EP4 expression, we have found many genes encoding the components of complex 4, complex 1, and complex 5 are downregulated in the EP4 high expressing cells. Okay, so those are the data marked that shows the downregulated genes in the EP4 high expressing cells. We also found that the EP4 expression is associated downregulation of a series of ribosomal protein genes. This is a large subunit, and this is the small subunits of ribosomal proteins. Quite extensive association. Okay, ribosomal proteins are regulated by the MYC, c-MYC genes, so the MYC is, so that suggests that EP4 associations may be linked to the decreased MYC activity. Okay, so the message one, PTGR4, EP4 expression in CD8 positive T cells infiltrating human cancers is positively correlated with expression of genes in recently activated T cells, such as CD44, CD69, AP1, and with that of some NF-kappa B components. On the other hand, EP4 expression is negatively correlated with expression of genes in IL-2 signaling nuclear encoded mitochondria genes, OXPHOS, and genes for ribosomal proteins. The last is linked to decreased MYC target, GSEA. Then what's about the myeloid cells that also express high EP4? We subclustered myeloid cells into 4, TAN PMN, TIM VCAN, TIM C1QA, TIM C1QA dividing. And again, the mapping, the COX1 and COX2 and EP2 and EP4 expression, as shown here. Okay. As indicated also here, EP4 expression is not expressed in all cells, but at most 50% of cells just expressed here. So we then carry out the EP4 high and EP4 low expression cells for comparison of the gene expression signature and extracted the gene expressions associated with EP4. Okay? And the GSE analysis showed that the upregulation of TNF-alpha signaling and the inflammatory response, but also showed the downregulation of the MYC genes and also oxidative phosphorylation here. Okay, EP4 association, EP4 expression is associated again the AP1 gene upregulation, NF-kappa B gene upregulation, but negatively associated with OXPHOSPHO and ribosome. And that is also seen in a series of those proteins. Okay, so message 2 is EP4 expression is negatively correlated with expression of OXPHOS genes and ribosomal protein genes in both T cells and myeloid cells infiltrating tumor. So the next question is that this association is just a mere association or causative relationship. to do that, we look into the data of our single-cell RNA-seq analysis of LLC1 tumor, in which we examine the effects of EP2 and EP4 antagonists on gene expression. In these experiments, we implanted LLC1 tumor cells subcutaneously in mouse day 0, and from day 12, we administered EP2 antagonists and the EP1 antagonists in combinations daily. And day 30.5 or 18, 1.5, and 6 days after antagonist administrations, we sacrificed mice and tumors dissected, dissociated to single cells, CT45 immune cells, FACS sorted, and subjected to scRNA-seq analysis. Okay, we then carried out the DEG analysis, differential expression gene analysis of each cluster between control and EP2I and EP4I treated mice. Okay. When we carried out the DEG analysis, we have found upregulation of oxidative oscillation and upregulation of the MYC targets. Consistent with this GSCA analysis, we have found upregulation of OX-phosphogene from in a variety of immune cells. That is the opposite phenotype we've seen in human samples. Okay? And that is also true, the ribosomal protein gene expression. Yeah. So EP2 and EP4 antagonists in combinations that made the reverse the phenotype of seen in human sample. Then is downregulation and the reversal of also genes related to mitochondrial function. It is functionally important, and what factor determined their expression. OX-phosphogenes is regulated in expression by the transcription factor PGC1. Okay? This transcription factor receives many signals, upstream signals, and that make a complex, for example, nuclear respiratory factor 1, and they facilitate the transcription of mitochondrial DNA, electron transport, oxidative phos genes. Okay? To examine the functional relevance, we treated LLC1 tumor-bearing mice with EP2I and EP5 from day 9 to 16. And we dissected the tumor day 17 and dissociated single cells, myeloid cells and T cells, FACS sorted. And we stained with TMRA to measure the mitochondrial membrane potential, and with antibody to PGC1 alpha, a master regulator of mitochondrial biogenesis. By doing that, we would like to look whether the EP2I, EP4 treatment increases the mitochondrial functions by raising mitochondrial membrane potential. Indeed, in these cells, you will see additions of the EP2, EP4I. In monocytes, the elevated the mitochondrial membrane potential, and that is also so tumor-associated macrophages and PD-1 positive CD8 T cells. And in these set of experiments, we have found that PGC1 alpha is the level is upregulated also the monocyte, TAM, and the PD-1 positive CD8 T cells. Okay. So message 3, in mouse LLC1 tumor model, administration of EP2 and EP4 antagonist in combination reverse downregulation of expression of OXPHOS genes and ribosomal protein genes in a variety of immune cells infiltrating the tumor and appears to reinvigorate these cells in tumor microenvironment. These findings indicated that PG2, EP2, EP4 signaling mediate immunosuppressive microenvironments by downregulation of OXPHOS and ribosomal protein genes and inducing their dysfunctional states in human tumors. So the next question is it direct effects of PG2 on immune cells? We tested these issues in CD8 T cells. We first found that the PTGR4 expression is induced in the CD8 T cells upon TCR stimulation. That's 8 hours, 16 hours, 24 hours. So this result is consistent with our findings that EP4 expression is associated with the activation markers and also the EP1. And we cultured the CD8 T cells stimulated with anti-CD3, anti-CD28 antibody and 30 units per meter IL-2 in the presence or absence of 30 nanomolar PG2 for 48 hours. We then washed and stimulated with or without IL-2 for 6 hours and probed for IL-2RA, for flow cytometry, and for c-MYC and PGR-alpha for Western blotting. You will see the PG2 treated shows the downregulation IL-2 receptor A, that's CD25, that's required for the high affinity IL-2 receptors. And when we stimulated those cells, we will see that the decreased the MYC staining in the Western blots and decreased PGC1 staining in the PG2 treated cells. And at 36 hours, we'll see here that IL-2RA downregulation is more marked, and that is corrected by the addition of the EP4 antagonist and EP2, EP4 antagonists in combinations. And that is also true for the mitochondrial membrane potential. You will see here those cells with decreased mitochondrial membrane potential is corrected by the addition of the PG2 EP4 antagonist. Okay? And due to the ribosomal protein deficiency defective in the ribosomal protein expression and OXPHOS protein expressions, PG2 treated cells cannot expand in this very critical period from 48 to 72 hours. Okay, so message 4. EP4 is expressed on T cell activation in CD8 T cells activated by PG2 if present, and impairs IL-2 signaling by downregulation of IL-2RA subunits CD25. Impaired IL-2 signaling can lead to attenuation of c-MYC and PGC1-alpha, thus possibly suppressing expression of ribosomal protein and OXPHOS genes. And clarification of mechanism in myeloid cells in progress in my laboratory. So in summary, 1, EP4 expression is positively correlated with activation marker genes such as AP1 in both CD8 T cells. I have not shown that, but the myeloid cells. Indeed, EP4 is induced upon T cell activation and CD8 T cells. EP4 expression, on the other hand, is negatively correlated with expression of a wide variety of OXPHOSPH genes in various immune cells in human tumors. And this downregulation and mitochondrial dysfunctions was reversed by administration of the EP2, EP4 antagonist to tumor-bearing mice. Consistently, EP4 stimulation with PGE2 downregulates expression of PGC1 alpha, a master regulator of mitochondrial biogenesis. 3. EP4 expression is also negatively correlated with the expression of c-MYC and expression of ribosomal proteins, both RPL and RPS globally. And this downregulation was also reversed by EP2 or EP4 antagonist in LLC1 tumors. 5. Our in vitro analysis suggests that the above dysfunctional phenotype is induced by a cell intrinsic mechanism of the EP4 action, at least in CD8 plus T cells. 5. These findings therefore indicate that PGE2-EP4 signaling in TME induces both energistically and proliferatively dysfunctional immune cells, and administration of EP4 antagonists can reverse and vitalize them to fight cancer. Okay, so this work has been carried out by the people of our laboratory of Department of Drug Discovery Medicine, Kyoto University, and the Shiracom has the major contributions of the work presented today. And I also thank various organizations for the research grants to carry out this project. Thank you very much for your attention.
Excellent presentations. Any questions?
Yeah, very nice work. And I'm Dizhu from Fudan University. I have two questions. So you just reported that there are EP1 expression in various of the immune cell. How can we learn that the mechanism is a major mechanism on the CD8 T cell? Could be whether there is a different cell-cell interaction, makes these things happen together. Yeah.
Are you asking what kind of the subsets of CD8 T cells?
Yeah. What's the driving mechanism? Because EP4 is expressed in various of immune cells.
Yes.
Whether the CD8 EP4 is a driving mechanism. Yeah.
We have not yet clarified the detailed mechanism of EP4 expressions, but it occurs after the T cell activations. So suppose it may link to the T cell signaling. And we think that this forms somehow a negative feedback mechanism. Okay? So T cell activated, and the EP4 is expressed, and when the PGE is there, it acts on the EP4 and downregulated T cell activation.
Yeah.
Okay?
Yeah, thank you. And so my second question is, it's very interesting to see the CD25 is the down expression in the T cell. Do you ever look at the Treg ratio, whether it's also reduced because the CD25 is a marker of the Treg?
You are saying that whether the Treg, regulatory T cells.
Yeah, regulatory T cells, yes.
Is also affected.
Is also affected, okay.
We didn't check it, frankly. We haven't checked it, but we think that it may also affect the Tregs. Yeah.
Okay.
But we have to check it.
Oh, great.
Okay,
great.
thank you very much for your question.
I think the EP4 inhibitor, the phenotype is very interesting.
Yeah.
Have you ever looked at the EP4 conditional knockout mice on the CD8 T cell,
yeah,
whether the CD25 is also downregulated?
Yeah. In relation to Treg, we examined the effects of the PGE2 in Treg in the LLC1 tumor. Okay? And in LLC1 tumors, EP4 stimulations activated Treg, not inactivated Treg.
Mm-hmm.
So the Treg might be different from other T cells. Yeah.
Okay.
But
Thank
we
you
have
very
to
much.
check. Thank you.
Yeah.
Fitzgerald in Philadelphia. Shuh, that was a great paper. A couple of questions. Were the patients from whom you got the samples on checkpoint blockade? You alluded to the interaction between the PD-1, PD-L1 pathway and the prostaglandin pathway. And I wondered if that made a difference, or indeed if there was a difference in the distribution of expression of the prostaglandin genes amongst the immune cells harvested from the 3 different tumors.
Patients we examined has not received any immune checkpoint blockers. Okay?
Okay.
That's one thing. And we presume that the EP4, EP4 antagonists can collaborate with immune checkpoint, the blockers.
Right.
Because you know that usually the T cells, activated T cells, utilizes the glycolysis as a major source. And the contribution of mitochondria is ignored. But if you vitalize mitochondria, it's enhanced activity of the immune checkpoint blockers.
Mm-hmm.
Yeah.
So a more speculative question. The CMYC observations were extremely intriguing. And as I'm sure you know, there's a bidirectional relationship between the molecular clock and CMYC in cancer. And I wondered, given that you had expression of the prostanoid genes in some cells, but not all cells, in the relevant clusters, whether the timing of sampling could have been something of relevance in terms of heterogeneity.
So we see that the EP4 expression is associated with cell activation. Yeah. So that's indicated that all the cells in tumor microenvironments are activated. That is also true for the T cells. Yeah, some T cells are activated and express EP4, but some T cells are not activated. So that doesn't express EP4.
Okay, thanks.
Thank you.
Any more questions?
Okay,
Thank you very much.
thank you.
So now it is my pleasure to present Lucie Clapp, professor of UCL Institute of Cardiovascular Science. She is also the deputy director of this institute, and she is fellow of the British Pharmacological Society and fellow of the Royal Society of Biology. Lucie, we can present your prostanoid receptor in pulmonary hypertension presentation.
Okay, I'd like to thank the organisers for inviting me to this World Symposium in Pharmacology. It's great to be back to face-to-face talks. And I'm going to be talking about prostanoid receptors in relation to pulmonary hypertension. How are they expressed? And really, what is the implication of activating different prostanoid receptors in pulmonary hypertension? I think up front I would like to thank my collaborators, particularly Dr. Shaheen Mardina from Great Ormond Street, Professor David Abraham from the Royal Free, and collaborations with Queen Mary, particularly Professor Brendan Whittle, and without whom I could not have done this work. Well, we don't need too much explanation, but Arachidonic acid is metabolized into the unstable prostaglandin E2, and various prostanoids are produced by the action of different synthases, and these are the receptors that thromboxane A2, prostaglandin F2 alpha, prostaglandin E2, prostacyclin, prostaglandin D2 activate. So if you activate these receptors, you get a rise in intracellular calcium, a drop in cyclic AMP, but also when you get a rise in calcium, you also activate the phosphodiesterase enzyme E1 to also decrease cyclic AMP, and this will cause vasoconstriction. And then when you activate these GS-coupled receptors, you get increase in cyclic AMP and vasodilation. So the consequence of activating these different receptors will become more evident as I go through the talk. I just want to remind you that if you contract pulmonary arteries with thromboxane, a mimetic, and then you either add PGE2 or PGD2 to arteries and veins, this is the profile you receive. So the contractile effects predominate to PGE2 in arteries, whereas the vasorelaxation response to prostaglandin D2 is very evident in veins. And we think that the contractile response to PGE2 is related to EP3, which can synergize with a number of contractile receptors to enhance the activity of these receptors. And this work has been done by a former postdoc in my lab, Nelson Ori. Now, some of the very earliest evidence that prostaglandin synthesis is disrupted, or prostanoid synthesis is disrupted in pulmonary hypertension, came from very early work showing that the urinary metabolite of prostacyclin is decreased in idiopathic pulmonary arterial hypertension and other causes of pulmonary hypertension. And at the same time, you get a rise in the thromboxane metabolite in the urine, particularly in relation to pulmonary hypertension associated with collagen vascular disease. Now, I haven't got time to explain too much about the pathobiology of pulmonary arterial hypertension, just to say that there are a number of risk factors, connective tissue disease, but also other groups of pulmonary hypertension are upregulated in response, group 3 pulmonary hypertension is related to a series of disorders related to hypoxia. There's a genetic component, particularly effects of the TGF-beta family, so you get loss of function of BMPR2, but you also get loss of function of this potassium channel task 1, and I'll show you some data about that. And these combine with other environmental factors to cause endothelial cell dysfunction, particularly downregulation of nitric oxide and upregulation of mitogens, including platelet-derived growth factor. And then you've got a change in the smooth muscle cell phenotype. So a downregulation of potassium channels, upregulation of proteins like TRPC6, which is a calcium, is a cationic channel allowing calcium to go into the cell. And then you've got upregulation of the phosphodiesterases, different types. And this all combines with the activation of, or the release of cytokines, the activation of NFAT and metalloproteases, combined to give you an aggressive vascular remodeling disease. And many arteries become muscularized and some become obliterated, and then patients will die of right heart failure. Now, prostacyclin was the earliest agent given to treat patients with pulmonary hypertension, and they were given to patients in as early as 1982. And it's a very potent dilator of the pulmonary circulation, and it will lower pulmonary arterial pressure, decreasing pulmonary vascular resistance, and thereby reducing afterload and increasing cardiac output. It's well known to be a potent antiplatelet drug, although the role of platelets in pulmonary hypertension is poorly understood. But also prostacyclin is a major inhibitor of smooth muscle cell proliferation. So prostacyclin is clinically very hard to work with. It has a very short half-life in vivo and is chemically unstable. So stable analogs of prostacyclin were produced, and this is what iloprost looks like with an increased half-life in vivo. This is what treprostinil looks like with a much longer half-life in vivo. So, but it took many other years to develop what are described as more selective and non-prostanoid type of IP receptor agonists. And in 2007, selexipag was developed as a selective IP receptor agonist with a much longer half-life, but it's a prodrug which is metabolized into an agent called MRE269 or ACT333679. And then very recently in 2010, we have relenipag, which it's an oral with a half-life of 24 hours. So over many years, we've been trying to understand, do these agents behave as just related to the IP receptor, or are other targets involved in their biological effects? So this is the receptor affinity against the human IP receptor. So you have prostacyclin, iloprost, and relenipag, very potent activators or binders to the IP receptor, high affinity. And at a 10-fold lower dose, you have agents activating the IP receptor, and then the selexipag is the prodrug, has a 10-fold lower affinity at the IP receptor compared to these agents. So one of the first things that we were able to demonstrate was if you were to measure cyclic AMP, this has actually been done in human pulmonary smooth muscle cells from patients, isolated from patients with pulmonary arterial hypertension. What you can clearly see is that the selexipag metabolite not only is 10-fold less potent, which is what you predict, but it gives you a lower maximal response to cyclic AMP, suggesting it behaves like a partial agonist. Likewise with relenipag, again, it in these assays seemed to behave like a partial agonist. Is this relevant to their biological effects? And this work was done by Dr. Lei Shen, a postdoc in my lab. So it's a bit more complicated than this because we also, myself, our lab, and others have shown that prostacyclin analogs will bind differentially to different prostanoid receptors at clinically relevant doses. So you have to remember that iloprost is a potent activator of the EP1 receptor, so it's equipotent with the IP receptor, and treprostinil is a 10-fold better activator of the EP2 receptor and the DP1 receptor. These are experiments where these human receptors were expressed in HEC293 cells. So what does it mean biologically? Well, if you were to contract small pulmonary vessels with a thromboxamimetic and look at increasing doses of treprostinil, it causes dilation or relaxation of pulmonary arteries, which is completely inhibited by the IP receptor antagonist. In vein, if you are under similar experimental conditions, you see that relaxation to treprostinil is only partially reversed by the IP receptor antagonist, suggesting that other receptors, in addition to the IP receptor, are involved in relaxation in veins. And a similar profile is seen in human pulmonary arteries. We think that some of this relaxation is related to the DP1 receptor. So an explanation perhaps for the differences in the profile There's little expression in small arteries of the DP1 and the EP2 receptor, whereas in veins, there's strong expression of the DP1 and EP2, and particularly in the endothelium. So if we compare relaxation to different prostacyclomimetics in small pulmonary arteries, we find that treprostinil and iloprost appear to be equipotent as well as generating similar maximal responses. However, if you compare the effects of ralinepag with MRE, these are the non-prostanoid IP agonists. Surprisingly, ralinepag becomes a full, it relaxes the arteries fully, but MRE only provides partial relaxation in these arteries. And so one of the experimental observations we saw is that some arteries just didn't respond at all to MRE 269, whereas other arteries clearly gave you quite strong relaxations. And so it's almost like this agent has an all or none effect. And we think that's probably related to either the activity of EP3 receptors or overactivation of phosphodiesterases, keeping cyclic AMP levels low and IP receptor activation low also. And just for comparison, we just show that iloprost and treprostinil relax somewhere in between the two agents. And so in terms of the difference between selexipag and all the other prostacyclomimetics, selexipag does not have any in vivo antiplatelet effects. And this is just data where patients were treated with selexipag for 3 days and compared to a placebo, and there's no decrease in ADP-related aggregation. So what might be going on? Well, we know that the selexipag metabolite does inhibit platelet aggregation in washed human platelets, but the dose at which it does is beyond that what you would give in patients. And so again, what I've just said before is we think that certainly when you get an all or none effect, it's probably related to the limitation of cyclic AMP elevation. So I'm going to switch now to studies in lung tissue and pulmonary arterial smooth muscle cells derived from patients with either pulmonary arterial hypertension or from group 3 PH. And this was work done fairly recently where we were looking at beroprost relaxation in control arteries versus those arteries taken from patients with pulmonary hypertension. So the first thing to note is that beroprost, as you would expect, is a good relaxator of preconstricted pulmonary arteries, but has little or no relaxation in arteries taken from PH patients. And in fact, in many cases, you see a small constriction. So what happens when you look at the expression of the IP versus the EP3 receptor? Well, we and many others have shown that the IP receptor expression is downregulated in PH. It's maybe difficult to see the brown staining disappearing in the diseased patient sample, whereas EP3 expression seems to markedly go up in PH. And also there are other studies showing that hypoxia increases the expression of EP3. So maybe that may be one reason why the expression goes up. In other experiments, people have shown in PH models that the sensitivity to EP3 agonists go up. So clearly we've got an elevated EP3 receptor activity in PH. Now, when you do the same experiments looking at ralinepag, we find little or no diminution of the relaxing responses in PH arteries, and these are small arteries taken from these patients. And this drug has completed its phase 2 randomized placebo-controlled trial and it met its endpoint with a significant reduction in pulmonary vascular resistance. And there's now a phase 3 study which is expected to be completed in December. And so their results will be eagerly awaited. So I'm going to, for most of the rest of the talk, talk about smooth muscle cell proliferation and some experiments in fibroblasts. So here are our two IP or non-prostanoid IP receptor agonists. Both of them inhibit smooth muscle cell proliferation stimulated by serum. But you can quite easily see that these effects are completely blocked by the IP receptor antagonist. So they are, to all intents and purposes, working solely through the IP receptor. However, if you look at the effects of iloprost and treprostinil, both of them inhibit smooth muscle cell proliferation with a slightly increased Emax. But the effects of treprostinil are relatively insensitive to the IP receptor antagonist. And then curiously, with iloprost, it's relatively insensitive to the antagonist, and then you get this reversal of the response. We don't think that's reversal or inhibition of the IP receptor. We think that's probably activation of the EP1 because you've got an enhanced contractile response at these higher concentrations. And we think that the EP2 receptors may be involved in the anti-proliferative response to treprostinil. So what's the evidence for this? Just first of all, I wanted to acknowledge Dr. Jishiga Patel, who did this work. Well, so first of all, we wanted to look at the prostanoid receptor expression in cultured smooth muscle cells, and this is the expression level in control cells. And the first thing to note is the IP receptor expression is similar to the EP2 receptor expression. And at least in culture, we rarely found DP1 receptor expression. So if you do the same experiment looking at message levels to these receptors in tissues isolated from PH patients, and these are smooth muscle cells, what you see is that the message level for IP goes down, and then the message levels for the EP2 receptor goes up. And so you're kind of driving potentially this receptor to become an activatable target in PH. And then this is just Western blotting data showing reduction of the protein, the IP receptor, and enhancement of the EP2 receptor. And so we wanted to see whether EP2 receptors were functional in these smooth muscle cells, and the answer to that is yes. So this is cyclic AMP generation in response to butaprost, a relatively selective activator of the EP2 receptors. And then when you downregulate the EP2 receptor, the cyclic AMP generation is almost abolished, and likewise almost abolished with the EP2 antagonists PF044. If you do the same experiment, now looking at cyclic AMP elevation with treprostinil, first thing to note is that the amount of cyclic AMP you get is higher than you get with an EP2 agonist. And then the cyclic AMP levels is only partially sensitive to removal of the EP2 receptor gene. So this suggests that treprostinil is activating cyclic AMP through more than one pathway. And so then if you look at the anti-proliferative response to clinically relevant doses of treprostinil, so here we have response to 10 nanomolar treprostinil and then a higher concentration, 1 micromolar. The majority of these effects are reversed when you knock out the EP2 receptor. And if you compare that to MRE, which we think is working entirely through the IP receptor, so that the smaller anti-proliferative response essentially is not changed by downregulating the EP2 receptor. So clearly we have functional evidence for EP2 receptors contributing to the effect of treprostinil. So I just wanted to show you just one or two bits of information about how do we think that the IP receptor and the EP2 receptor might be working to inhibit smooth muscle proliferation. And also because with loss of function mutations in task 1, it's really the idea that perhaps in those cases, maybe we can rescue either the activity or expression of task 1. So what we noticed was that treprostinil, when you treated smooth muscle cells with it, there was a significant increase in the expression of task 1, and a trend when you either stimulated cells with the IP agonist or the EP2 agonist. And this work was done by Jerry Abdu-Hamda, who's a PhD student in my lab at the time. And then if you overexpress EP2 receptors with task 1, you can see that treprostinil increases the current density of this particular potassium channels. So this is evidence that perhaps we should be giving treprostinil in those patients with loss of function of task 1. And then these are still experiments looking at the task 1 inhibitor, showing that the anti-proliferative response to MRE is certainly reversed in these experiments, with the agent on its own having no effect on cell proliferation. Likewise, with butaprost, the anti-proliferative effects of butaprost are reversed more or less with this inhibitor. Obviously, with all pharmacology, you'd want to be able to look at this with somewhat more selective agents or other ways to show this to be correct. And then if you look at the effect of the task 1 inhibitor, it certainly reverses partially the response to treprostinil. So I'm just going to show you another difference between treprostinil and the selexipag metabolite, in that when we isolate fibroblasts from patients with pulmonary arterial hypertension, what we find is that treprostinil is able to inhibit proliferation of fibroblasts, whereas the IP-selective agonist MRE-269 was very weakly inhibiting cell proliferation. So, and then just to note that the EP2 receptor antagonist is able to inhibit partially the response to treprostinil, suggesting that EP2 receptors are involved in the anti-proliferation response. So what about other evidence for EP2 expression and IP receptor expression in other pulmonary diseases? Well, so in group 3 PH, this is a collaboration with Xavier Norel, certainly the ratio of EP2 to IP goes up in group 3 PH, although this is more related to a reduction of the IP receptor expression than enhancement of EP2. And also it's interesting to note that in asthma and in patients with chronic obstructive pulmonary disease, you do see an increased expression of EP2 receptors, a heightened sensitivity to prostaglandin E2, suggesting that certainly this receptor is upregulated. And then this is really showing evidence that treprostinil may actually reduce the transition of a fibroblast into a myofibroblast. And these are fibroblasts taken from a patient with idiopathic pulmonary fibrosis. And what you notice is that the cells under basal conditions are already partially transformed. They express alpha smooth muscle actin and also F-actin. If you stimulate with serum, you increase the production of collagen as well as enhance the expression of the agents that drive the transition into myofibroblasts. If you give the transforming growth factor here, you get a huge upregulation in collagen production. And treprostinil is able to inhibit both the effects of serum as well as the effects of transforming growth factor. So we think that this agent is potentially also working as a potent antifibrotic agent. And this is really interesting data because this is a high put-through screen where the transforming effects of TGF-beta in driving myofibroblast phenotype. What was identified in this screen was that both the EP2 and the EP4 receptors were amongst the most potent, if not the most potent targets to inhibit pulmonary fibrosis. And I just want to finish with just one or two slides. One showing, what about the role of the DP? I've talked a lot about EP2, and this is recent work showing that the DP1 receptor may be involved in, has a sort of protective effect against vascular remodeling and also the transition from a smooth muscle cell phenotype into a myofibroblast. And so this is just a model of pulmonary hypertension using hypoxia and Shuh, and sort of under control conditions. This is the right ventricular systolic pressure. It goes up in the presence of hypoxia and Shuh, but is markedly elevated when you knock out the DP1 receptor, and also remodeling is much stronger in the absence of the DP receptor. And then some other work by these colleagues showed that if you knock out the DP1 receptor, you drive the phenotype of smooth muscle cells into a myofibroblast. So I think my take-home message here is that we have an agent which is not only an IP agonist, but is also a DP1 and EP2 agonist. And very recently was subjected to a clinical trial in group 3 PH and was found in this group of patients, so those with interstitial lung disease, where a lot of them will have pulmonary fibrosis, essentially they were able to see some benefit in these group of patients. And we think this is related to the unique pharmacology of treprostinil and other agents in this group actually failed failed in group 3 PH. So I'd just like to summarize what I've hopefully shown you, is that clearly we started with a group of agents which we thought worked primarily through the IP receptor, but we now know that there are a heterogeneous class of compounds activating a variety of different prostanoid receptors. We know that these two agents, they're non-prostanoid agents, but they act as a partial agonist, and they essentially do work almost entirely through the IP receptor. And I guess this begs the question as to whether drugs that just target one receptor versus drugs that perhaps activate more than one receptor potentially have superior benefit. And that really is going to be the ongoing question clinically. What we've shown is that there's robust EP2 expression, a downregulation of the IP receptor, and probably a downregulation of the DP1 expression in pulmonary vascular remodeling diseases. But these clearly are important targets clinically and involved in anti-remodeling processes, and that we think that task one is clearly involved in regulating regulating the cell proliferation in smooth muscle. And I've mentioned some of them before, but here are the acknowledgements of my collaborators. I've obviously acknowledged the people in the lab that did the work. And these are some of the funders. Thank you very much.
Okay, thank you, Lucie, for this quick talk. It is now open for discussion.
Hi, thank you for a great talk. I was quite interested to see the all-or-nothing response in the human isolated vessels.
This is to the MRE 269. Yeah.
So is that seen in the patients with pulmonary hypertension?
Yes,
That
actually it is.
And then I guess going further down there, have you been able to dissect the kind of reason for the non-responders?
So I think it's one of two things. One is that some patients, you see very little IP receptor expression, so that's going to be one problem. I think in other patients you may get, particularly those that are where the lung is hypoxic, you may get an upregulation of the EP3. And those two things mean that the MRE 269 metabolite, the successive metabolite, essentially isn't able to increase suctic AMP to the extent that, say, a full agonist might.
So the full agonists still have an effect on some of the non-responders?
Absolutely. So I didn't show you the data, but if you look at non-responders to MRE, they still respond to all the other agonists. So I think what we're going to have to do is almost generate a screen in those patients, because selexipag is an oral, so it's easier to use than some of the other administration routes like subcutaneous or intravenous. So I think it's very important that we actually understand who's going to respond and who won't.
Thank you.
Yes, please.
Thank you very much. That was lovely. May I just ask a slightly technical question? Early on, you mentioned you'd measured the affinity of various compounds in HEK cells.
Yeah.
Could you just tell me what assay that was and how you did it?
They were sent off to Syrup. So the French company who did the binding assays.
Okay. Do you know if it's radioligand binding or?
Yes, it was.
Okay.
Yes, yeah, no. So I mean, so what they were competition binding experiments.
Okay. Yeah.
Yeah.
Okay. Done by Sarah.
Yeah.
Thanks.
Just one question about the affinity of these prostacyclin analogs, ralinepag, and is without any other selectivity for other prostanoid receptors? Absolutely. But
So ralinepag, although in all our assays appear, certainly in the cell proliferation assays, seems to work entirely through the IP receptor. But my feeling is in the relaxation studies, there has to be another mechanism. But we don't know necessarily what that is, because we were very curious as to why it gave you full relaxation of our pulmonary arteries.
yes, please.
Hello, Urs Röke from Geneva. You showed a slide that showed that from different pulmonary arteries from different patients reacted very different or to different levels, and you argued that it might be due to the different levels of cyclic AMP. So there you could add PD-1 inhibitors, for example, to level that out. And the second question, or related question, why are not PD-1 inhibitors used in pulmonary hypertension?
So in answer to your last question, I'm not sure, although it's a bit more complicated than that, because PD3 and possibly 4 are also involved in regulating cyclic AMP generation to these agonists. But I personally think that PD1 is an important phosphodiesterase because it's activated by calcium, so it's going to possibly be the one that has the most activity. I'm guessing that probably PD1 inhibitors, maybe they're toxic in humans, but I think there's a real risk here because people looked at milnarone in trying to elevate the activity of these compounds and found that was deleterious, and that might be because if you have heart failure and you have a lower systemic pressure, you might actually, they may be contraindicated. So it's a really complex thing to answer. But I get your point, and I think it would be really interesting to look at different PDE inhibitors. But doing these human artery experiments has taken a long time because it's a rare disease, and it's taken us years even to generate all this data.
Yes, thank you. Milrinone also failed in the heart failure trial because it made people feel better but die earlier.
Yeah, I mean, it could be the heart's pumping too hard to overcome systemic lower pressure.
Yeah, okay.
Okay.
Great.
Thank you, Lucie. We have to move to the next presentation.
Okay, thank you, Lucie. So our next presentation is Xavier. And Xavier is from the French National Institute of Health and Medical Research. He led the group of Eicosanoids and Vascular pharmacology. He's a fellow of British Pharmaceutical Society and the chair of the NCIU-FAR Prostaglandin Receptors Subcommittee. Yeah, and welcome.
Thank you, Chengcan, for these words. I will present, my talk will be about the receptors of PGE2, which are involved in the vascular system and also in vascular physiopathology. This work that I will present have been performed at the Bichat Hospital and in Paris, and they are most of them are published, but some are unpublished data. I will start with this biosynthetic cascade of the PGE2, as you have seen before, and COX is involved, and PGS1 is involved, and after PGS2 produced will act on 4 major subtype receptors, which are the EP2 and EP4, which are coupled to the GS protein to increase the CAMP and to induce vasodilatation when it is expressed on a smooth muscle cell, and also to be anti-proliferative. And the 2 other receptors, EP1 and EP3, this one will stimulate an increase of intracellular calcium. And also for the EP3, there is a decrease of CAMP, an inhibition of adenine cyclase. So I have also highlighted here the TP receptor, because we will see in the presentation that this receptor also is linked to PGE2 activity. So why to study the PGE2 in cardiovascular system? For 2 reasons. First one is that most of the pathology, cardiovascular pathology, are involving inflammation, like in atherosclerosis or in aneurysm. And in this case, there is COX-2 induction, and PGS1 also induction. And as is in this model of acute inflammation that we have done on human memory artery, we have seen that the incubation of these vessels for only 6 hours or 24 hours induced COX-2 expression, of course, but also but this model is using interleukin-1 beta and LPS. And after incubation with these compounds, we got an increase of 2-fold of the production of PGE2 here, and a 4-fold increase of this production after 24 hours. A similar result was observed with the stable metabolite of prostacyclin, this keto-PGF1 alpha, as you can see here. And also the thromboxane was not affected by the by the inflammatory conditions, as you can see here. So the second, the second reason to study prostaglandin E2 is that there are some prostaglandin E2 and some of its analogs are involved in clinical practice, like in gynecology or in gastrointestinal disorders. And these compounds used are the following: PGE2 as dinoprostone, misoprostol as cytotec, and suprostone as nalador. And I also here I mentioned the affinity of this molecule for the different EP receptor, as indicated in the UFAR BPS guide to pharmacology. As you can see, PGE2 could act on both EP, all the EP, of course, but EP3 and EP1 with a little selectivity for EP3, but a small one. And while misoprostol is mostly more selective for EP3 only, no activity on EP1. And sulproston, this one is strongly has a strong affinity for the EP3 receptor, as you can see here, and a lower activity for the EP1, while the 17-phenyltrenor PGE2 is quite equal in affinity for EP3 and EP1 receptors. That means that in the next slide that I will present to you, when we have a huge difference in the relaxation, dose-dependent relaxation that we have induced between sulproston and this agonist, that means that there is the involvement of an EP3 receptor, and when the two curves are similar, that means that it's more EP1 response. So, as an example, I will show you the result we have obtained in an organ-based system with isolated human mammary artery in this colon and human saphenous vein in this colon. These vessels are used for coronary bypass and surgery, as you can see here. That is mammary artery. The saphenous vein are here. That is legs veins. And on the first line, it is protocol that is more suitable to study contractions. It is on basal tone, and as you can see, all the first PGE2 induced contraction is a black circle curve here, while all the other agonists, which are most of them have selectivity for the EP3, are inducing contraction, and sulproston and 17-phenyl PGE2 are, there is a huge difference between the two dose response curve, suggesting the involvement of the EP3 receptor in the contraction in the mammary artery. In contrast, in the human saphenous vein, these agonists were giving very few small contractions. Only sulproston gave a strong one, but this contraction was blocked by AGTE and calcium-free medium, suggesting the involvement of the TP receptor. So for the relaxation protocol here, that is pre-contracted vessels with norepinephrine. And as you can see, the two selective agonists for EP2 and EP4 receptor, which are the best candidate for relaxation in the vessel, were completely ineffective. And only as a positive control, we have the response here for the IP agonist, the hyloprost, giving a strong relaxation in this human mammary artery. In contrast, in the human saphenous vein, the selective EP4 agonist, ONO 329 here, give induced a dose-dependent relaxation, potent one, and also PGE2, the black circle here, induced a potent relaxation. The hyloprost also induced a potent relaxation, while the other selective agonist for the EP2 receptor only gave a response at a higher dose, and suggesting it was not selective for the EP2, it was a response through the EP4 receptor probably. So in summary, that we have only the EP3 receptor responsible for contraction in human mammary artery, and I will say only the EP4 receptor for PGE2 responsible for relaxation in the human saphenous vein. And we confirm this result with RT-PCR result data. As you can see here, the mammary arteries, the saphenous vein, there is no EP1 mRNA in 2 patients. There is EP3 receptor mRNA in these patients. And for the saphenous vein, only EP4 mRNA was detected. So that is what happened with exogenous prostaglandin and analogs. And here we have the involvement of the prostanoids in these same vessels, mammary artery and saphenous vein. The involvement of prostanoid in the vascular tone of this vessel during normal condition and inflammatory condition. It is the same model as previously with acute inflammation with interleukin-1 beta and LPS. And as you can see, in both cases, we have a significant decrease of the reactivity of the vessel in this acute inflammation, which is representative of a septic shock, for example, where we can see a blood pressure, a decrease of the blood pressure. So to make a link with the previous slide, with the EP3 receptor in mammary artery and the saphenous vein, the EP4 in the saphenous vein, This decrease here also could be explained maybe by decrease of the EP3 receptor expression, or either by the dominant effect of the prostacyclin. The prostacyclin is largely produced in this case, and that could be also explain this decrease. Same thing in the saphenous vein. The prostacyclin dominant effect could be also explain this effect, but could be also explained by an increase of the EP4 receptor now in the saphenous vein in the inflammatory conditions. Now I will shift on the other vessels, on the pulmonary vessels, and that is a previous work from the group of Bob Jones in Hong Kong in 1994, published in British Journal of Pharmacology. It was the first one showing the involvement of the EP3 receptor in the control of the vascular tone. As you can see, PGE2 was inducing dose-dependent contraction of the artery, and also they have also contraction induced by sulprostone and this at 17-phenyl PGE2, and with this huge difference between the two curves, suggesting also here that the EP3 receptor is present and responsible for contraction of PGE2 in the human pulmonary artery, as well as in the human mammary arteries that we see before. About the pulmonary vein, also, we have found that that is large pulmonary veins. That is not the same as Lucie Clapp presented just before. These ones are more responsive to PGE2 inducing relaxation of a contracted pulmonary vein. And this effect was blocked, inhibited, dose-dependently also, but here it is one dose, 1 micromolar of this EP4 selective antagonist, GW627. And we confirmed this result with two selective agonists, the ono compound here 329, with a selective agonist for the EP4 receptor inducing vasodilatation, and also with another EP4 agonist, which is the Merck compound, the L902, and both were were blocked, were inhibited, shifted on the right, with in presence of the GW, the EP4 antagonist. So this suggests result, as we have seen in the human saphenous vein before, that the human pulmonary vein, PGE2, is able to induce relaxation via the EP4 receptor of this, we say, medium and large vessels, which are at least more than 2 millimeter diameter. I want to specify that also there is not only the vein are showing that the EP4 receptor could be stimulated. Also, some arteries have been described in the literature. Urinary, renal, and cerebral artery are able to vasodilate due to the EP4 stimulation by PGE2. I will finish with the pulmonary vein with this result. This is just a comparison between human pulmonary artery and human pulmonary vein. We confirm that the EP3 mRNA is strongly more expressed in these arteries than in the vein. But we have seen also that the EP1 receptor was more produced in the human pulmonary vein, and we have also found that in the organ-based system that the Ilopros, which is not only IP receptor, it has some affinity for the EP1 receptor, and this Ilopros was able to induce dose-dependent contraction of the human pulmonary vein, and this result was confirmed by this one, where we have used the sulprostone in presence of the TP antagonist, the thromboxane receptor antagonist, and sulprostone is acting on both, first with a greater affinity for the EP3, but also on EP1, and here we have probably a response of sulprostone on the EP1 receptor, since this selective EP1 antagonist was blocking this response, this contraction in dose-dependent manner. Globally, that is suggesting that the EP1 receptor is present could be stimulated by PGE2 in the vein and human pulmonary vein, and to be involved in this contraction. Now I will show another side of this EP receptor in case of pulmonary disease, that is a COPD disease, chronic obstructive pulmonary disease. And we have tried to look if there was any difference in the expression of the different receptors. finally, we found no difference for all the receptors in the human pulmonary arteries, in these pulmonary arteries that we have used. Similar result was found also with the human pulmonary vein, just a tendency here, a little bit increase, but it is not significant globally. In this vessel of COPD patients, there is no difference between versus the control. That is in contrast with another pathology, that is aneurysm, human abdominal aneurysm, and that is a dilatation of the aorta, and there is a thoracic aneurysm, that is also abdominal aneurysm, and it is a work of a Japanese group, which they have shown that there is a strong, significant increase of the expression of the EP4 mRNA in the vascular wall of the aorta of these patients. And they have shown also that the gelatinase activity was different, since here you have the MMP9 expression in the biopsy of the aneurysm patients in the aorta, and not in the control cells derived from aorta, from patients without aneurysm. And the MMP2 is also expressed in both type of samples. And this metalloproteinase, matrix metalloproteinase, were under the control of the EP4 receptor, since here you have the use of the selective agonist for the EP4 receptor, and there is an increase, significant increase of the MMP2 activity, either on the cell derived from aorta with aneurysm, and also in the biopsy of this aorta, similar significant increase of the MMP2 activity. MMP9 activity was not changed after simulation with the EP4 receptor. Interleukin 6 was increased in both samples, cells, or the biopsy of the aorta, of the sample of aorta derived from the patients with aneurysm. So in this case, we have a strong effect on the development of the pathology due to the activation of the EP4 receptor by PGE2. In this slide, here is another vessel. It is human intercostal arteries that are derived from the aorta. You can see exactly the same thing that we have seen with the human mammary artery, the same thing we have seen with the human pulmonary artery. All the selective agonists are used, are the same. So Prostanoid is here. 17-phenyl are the square, or the curve with the square here. And we have the huge difference, and so suggesting the EP3 receptor is involved. The selective EP1 agonist here is not working. The ilopros is inducing only relaxation, so suggesting there is no EP1 involvement. And also we have used some antagonists on the PGE2 induced contractions. The black point are the curve induced by PGE2, the control curve, and with the Merck compound, the L826, that is a EP3 selective antagonist, we have obtained a significant decrease inhibition of these contractions, while the two selective EP1 antagonists were not changing significantly the curve. So here, similarly, as the two previous arteries, we have the involvement of the EP3 in the contraction induced by PGE2. And that is voilà. That is in addition to these results that I'm showing to you, done in the group. We have also preliminary data result on carotid, subclavian, and brachiocephalic arteries. All these arteries coming out from, derived from the aorta or the heart, are able to contract by, via the EP3 receptor stimulated by PGE2. The EP3 receptor is more complex than it looks like. In fact, that is one of the, there are three receptors in the human, in the prostanoid receptor in human, which could have isoforms due to splicing variant of the gene. And the genes is here. You have 10 different exons, and you could have a lot of possibility to have different splicing variant at the level of mRNA. Here you have a lot of mRNA, different mRNA also, but there are properties that is not exhaustive. But in bold here, I have also put in bold only the mRNA which were able to give a protein expression at the end. And the three blue here, that is the same mRNA. Not the same mRNA, that is they will give the same protein, this one with 390 amino acid. That is one isoform, proteic isoform, it is EP3-1. And there is also EP3-2, EP3-3, EP3-4, and EP3-E, which are the 5 human proteic isoform of the EP3 receptor today described. And they are variable at the C-terminal side, and there could be have 20 to 50 different amino acids in this region. And this difference could account for different internalization of this EP3 receptors. There is a publication from Bilson and al. It was in Febb's letter in the 2000, I think, showing that the EP1 was protein was completely after stimulation intracellular, going intracellularly, while there was only a partial after stimulation, a partial entrance of the receptor in the cell, and there is no entrance of after stimulation, no return of the receptor in the cell. This study on the EP3 isoform are quite rare. There are, I will show you the 3 that I have found. Maybe there are some other, but I haven't very easily found. And here you have, that is our result in human pulmonary artery and human pulmonary vein. As you can see, the 5 isoforms, that is only one, EP3-1, and also the 4 other isoforms. And as you can see, the black bar are the isoform detected in pulmonary artery, the gray one in the pulmonary vein, and each time, with the exception of the EP3-2, each time the EP3 mRNA isoform expression is greater in the artery than in comparison to the vein, and that is confirming the result we have shown before with the physiological data. Before to show you other example of this expression of isoform of the EP3 receptor, I want to show you this slide that is human coronary artery with or without atherosclerosis. That is without atherosclerosis, and this one is a patient with atherosclerosis. That means this coronary artery, we have dissected only here the supple party of the artery, because there is a lot of plate and so on. For organ-based study, it is not easy to work with a ring with plate inside and so on, plaque inside, and we have used only the supple part. Here there is that is non-atherosclerotic vessels. As you can see, the maximum contraction induced by PGE2 was about 350% here, and while here it is only 250% of the potassium chloride pre-contraction, and suggesting that either the EP3 receptor is decreased in the case of atherosclerotic coronary artery, or all the mechanistic of the contractiles, mechanistic of the smooth muscle is deficient in this coronary artery. In fact, we have made the measurement of the different isoform in this coronary artery, as you can see here. And EP3-1B was quite similar, with a little tendency at the decrease. But the EP3-2 is significantly decreased in coronary arteries. The EP3-4 is also significantly decreased in the coronary artery, while the EP3 is not significant and TP is not also different. So this decrease, at least these 2 decreases of these 2 isoforms, could account for the lower reactivity we found in the atherosclerotic vessels, coronary artery. Now I have a last slide from a group from London. This one we're working on another pathology, on aneurysm, also on the abdominal aneurysm. And as you can see here, they have shown that the EP3, 1B, 2, and 3, and 4 were present. And there was some difference between, they have used only one control patient and 2 patients with the pathology. At least there is one bond, another bond, which is not found in the patients. That is biopsy, and that is cell culture of the aorta vascular wall, of the wall of the aorta. And there is a difference here, and there is also a bigger bond here in this. But that should be maybe verified with more patients. So in this case, as well as in the previous slide with the coronary and atherosclerosis, we can see that the isoform could be interesting to be studied and to check if there is a variation between pathologic and non-pathologic vessels. I will continue, now I will come back to the coronary artery to show you that that is in link with the slide with also the isoform in the coronary artery, because that is coronary artery also with, that is ILC coronary artery, and that is pathological when you with atherosclerosis. And as you can see, all the enzymes responsible for PGE2 synthase and as well as the PGE2 production were increased. In each case, you have an increase of the enzyme and the PGE2 production. And maybe that could be also account for the what we have seen with the EP3 isoform, which are reduced in the coronary artery of the patient with atherosclerosis, because before there have been a lot stimulated with PGE2, and there is internalization of the receptors. And in this case, we understood why the EP3 isoform mRNA was reduced, and also why the contraction was reduced in this coronary artery of atherosclerotic patients. Now I will show you some data about physiological response of this coronary artery. That is ILC coronary artery, this one. And you have these vessels are very strange because in comparison to all the vessels that we are using, this one is giving a very potent contraction without a plateau, as you can see here. All the vessels you have seen before, there was plateau, but here there is no plateau. And in addition, after 1 or 2 hours of experiments, these vessels are unstable. They are giving vasospasm, and we have to work quite rapidly to avoid this vasospasm. But anyway, that is a control curve without treatment. And here you have, after treatment with a selective TP, the thromboxane receptor antagonist, the BU305, B4 or 5, we have a reduction of at least 50% of the contractions, and that this reduction was furthermore increased in presence of 2 selective EP3 antagonists. So the contraction induced by PGE2 in the human coronary artery, ILC1, is dependent of 2 receptors. The EP3 and TP receptor are involved in these contractions, and that is. We have a problem with that because, as you have seen maybe in the second slide, there was a TP receptor have very small affinity for the PGE2 has a very small affinity for the TP receptor. It is the pKi is about 4.5. It is very small, and maybe that is other effect, that is heterodimer or synergies between the EP3 and TP receptor, as it has been described in the guinea pig aorta by David Woodward and Bob Jones in the British Journal of Pharmacology 2011, I think. And that is probably, that is not an additional effect, it is a synergistic or heterodimer involvement. So now I will go to the effect of the PGE2 and which are its analogs, misoprostol and supprostol, which are used in clinical practice in pregnant women. And for this reason, we tested misoprostol in our system in the organ bath with the coronary artery. Here you have misoprostol data. It is in 8 patients, and in the same patient, 5 of these 8 patients were used also for PGE2. We find again the same results. That is the TP antagonist reducing by half the contraction, but we found exactly the same result with misoprostol, suggesting that in this coronary artery, the misoprostol is also very potent at inducing this huge contraction and due to the activation of both receptors, EP3 and TP. And this result should be maybe associated and linked to this huge literature about the PGE2 misoprostol or sulprostone cardiac side effects, that is myocardial infarction, that is coronary vasospasm, that is cardiac arrest, and that is not exhaustive. Maybe I could have 2 or 3 slides with all these side effects. So suggesting that maybe the use of this compound should be very maybe carefully used with different dose, but also maybe other compounds will be better to be developed. To finish these results, that is a result of a group from Los Angeles, and that is crystallography, this is a recent paper, showing the place in the plasma membrane of the EP3 receptor, the human one, that is a isoform 1, EP3-1. And as you can see here, you have the inside in green, the misoprostol in this binding pocket here. And what they have shown also that there is an extracellular loop 2, which is a red ribbon is here, which is coming to make a lead, blocking, closing the door, and the misoprostol stay inside and have very difficulty to go outside of this receptor after. And that could also an explanation for the side effect that I presented in the previous slide. So in conclusion, this of this presentation, that is EP3 receptor is frequently found in all these vessels in the artery, and when it is stimulated by PGE2, it will induce vasoconstriction. Mostly of these arteries derive from around the heart and the aorta. For the PGE2 could also activate EP4 receptor on some veins to induce vasodilatation, but also on some arteries to induce vasodilatation. The human vessels, when the contraction is induced in presence of inflammatory condition or pathological condition, when these contractions are reduced, that could be due to a prostacyclin dominant effect or a reduced EP3 expression or an increase of the EP4 expression. And finally, the PGE2 analogs used in clinical practice are activating 2 receptors, EP3 and TP, and which are very potent together, and could be not additive effect, could be synergism or heterodimer. And this potent vasoconstriction could account for the numerous cardiac side effects reported in pregnant women. I would like to thank all the people in Paris, mostly my student, my PhD student, my previous PhD student, and also all the people involved in collaboration and in the world. I will finish with this slide with a rapid advertisement for our meetings. That is a French meeting, not an European meeting at the Pasteur Institute about apoptosis and inflammation. That is our meeting in Edinburgh next year in June about bioactive lipids with Schengen. And here we have the WCI in 2024 about the Congress of Inflammation in Quebec City. Thank you for your attention.
Thank you, Xavier. Very nice talk. And any questions? Yeah.
So I was just curious with the results in the aneurysm studies, where you had an elevation of your EP4 receptor expression. Is there any evidence that maybe EP4 antagonists might be used to kind of reverse or maybe slow down the kind of aneurysm process, or is there any evidence in animal models that might?
I think this paper was quite complex. There was a lot of data inside, and it is not only paper showing this EP4 involvement in the aneurysm, smooth muscle cells, and probably the antagonists could be have been used in this the GW compound other EP4 antagonists have been used in these studies.
And then was the upregulation of metalloprotease 2, is it just specific for 2, or did it affect, I couldn't, maybe I missed it, did it upregulate MMP9 and...
No, the EP4 receptor activation was not inducing any effect on the MMP9. Just MMP2 was regulated by that is the work of the Japanese group.
Hi, thank you so much for the brilliant presentation. I was wondering, do we know there's any evidence for sexual dimorphism of those group receptors, given the response in the vasculature you've seen?
You mean between sexual man and woman?
Yeah.
No, I don't know. I don't know nothing about that. Sorry.
That's all right. Cool. Thank you.
Okay, any more questions? Yes, thank you very much, Xavier.
It is my pleasure to introduce Chengcan Yao, which is from Edinburgh University. He's a reader at this university. He was trained for his PhD and postdoc in Kyoto University with Professor Shuh Narumiya, and he is a fellow of the British Pharmacological Society, and also he is a member of the nomenclature, the UFAR nomenclature board subcommittee about prostanoid receptor. Chengcan.
Thank you very much. Thank you for your introduction. So I'm going to talk about the cellular-based Prostaglandin E2 function in the intestine. The gut is one of the largest organs in our body, and it contains about 70% of immune cells. And it has 10 times more microbial cells than the human cells. The gut, the interactions among the microbial, the epithelial wall, and the laminar propria immune cells determines the fate of the gut, whether it's health or diseased. So the immune system, especially the lymphocytes, so which the functions decide the gut homeostasis or disease, and we know that there are several subsets of the lymphocytes. So we have the adaptive lymphocytes. So I only show here T cells. So we have a regulatory FOXP3 positive regulatory T cells, and we have the Th1 interferon gamma or TNF producing, and also IL-17 producing Th17 cells. So this each different subsets of T cell, they have different functions. And for the opposite to the adaptive T cell, in our body, we have also innate lymphoid cells. So they like T cells, but they just don't have T cell receptors, such as IL-10 producing the regulatory kind of IL-Cs, and also the type 1 IL-C and type 3. So mainly we know that now that regulatory IL-10 producing the lymphocytes, they maintained the gut homeostasis, but also the IL-22 producing group 3 innate lymphoid cells or some Th17 cells, homeostatic Th17 cells. They also, through producing IL-17 and IL-22, to promote the gut homeostasis. This is because the gut epithelial cells is highly expressed IL-22 and IL-17 receptors. And in contrast, especially interferon gamma producing T cells, including Th1 cells, or some interferon gamma producing Th17 cells, they are pathogenic cells. So they drive the inflammation and even tumor development. So to study the gut health or disease inflammation, so there are lots of different animal models. So we could simply just divide them into two kinds of models. So one kind of model is that to use toxic chemicals or infections or food allergens or some transgenic models. So the first kind of heat came from outside of the gut that directly damage the gut epithelial wall and then induce the immune dysfunction or activation and leading to the acute gut damage or an inflammation which could mimic the human ulcerative colitis, food allergy, or infections disease. Another kind of model could be that such as a T cell transfer or immune activation, or some genetic models such as IL-10 knockout mice. So this model is basically the first heat. It came from inside of the gut. That means from the laminar propria area. So immune activation is the first event, and then the activated immune cell produce inflammatory cytokines which damage the epithelial wall and leading to basically T cell mediated chronic inflammation, so which could mimic the human Crohn's-like disease. So in these different models, we hypothesize that prostaglandin maybe have different function, that like prostaglandin maybe in different human disease, they have different functions. So I will skip this because Professor Narumiya and Xavier have already introduced the prostaglandin. So the gut is if not the only, but if not only, but probably the one of the more organs that produce highest prostaglandins. This is measured from the gut tissue, dry tissue, and then we can see that lots of prostaglandin produced endogenous in live mice gut. And when we use the COX inhibitor, such as in a medicine, a couple of days, and then can totally block the production of endogenous prostaglandins. And also the gut highly express prostaglandin receptors, especially the EP4 receptor, and at the whole tissue level. So for a long time, that it had been known that if we block endogenous prostaglandin production, such as by NSAIDs, or we can have some side effects in the gastrointestinal tract. And also, if we inhibit the prostaglandin degradation or activate prostaglandin pathway, such as the EP4 pathway, and then we can protect against gut damage. So by single-cell sequencing, then we have known that the gut the cellular-based expression of prostaglandin receptors. So we see that the EP1 and EP3 is barely expressed in main gut cell types, while EP2 and EP4 is highly expressed in in epithelial cells or immune cells, including myeloid cells and T cells, but not in this database data set. We cannot see the B cell express EP2 or EP4. So it has been published by other groups, several groups, that they suggested that PGE2, through acting on the EP4 receptor on epithelial cells using either conditional knockout mice, and to demonstrated that the PGE2 EP4 signaling in epithelial cells is important to promote epithelial homeostasis and protect against injury, while the PGE2 signaling in the immune cells, called the PGE2, both EP2 and EP4 highly expressed in immune cells, while not clear. So we tried to understand what's the PGE2 function in immune cells, regulate gut in the gut. So we used the first question we asked whether PGE2 signaling in T cells is important or affect the gut damage. So we used DSS-driven colitis model, so an acute DSS directly damages the gut wall, and you can get inflammation after a few days of treatment. Unfortunately, we couldn't see any evidence that for PGE2 signaling in T cells, it can have a protective or inflammatory functions in DSS-driven gut injury. We can also see that the PGE2 signaling in T cells did not affect either regulatory T cells or pathogenic Th1, Th17 cells in the gut. So then we asked whether PGE2 signaling other immune cells. So in this work, we simply treated the live Block 6 mice with LPS, followed by a few days of medicine treatment. So we can see that, as expected, that medicine treatment enhanced LPS-induced systemic immune response, and then such as splenomegaly, the cytokine production or neutrophil recruitment. And this was nearly abrogated in mice treated with antibiotics, and suggested that the gut bacteria or bacterial translocation may be contributed to COX inhibitor-dependent augmentation of systemic inflammation. And we also used RAG knockout mice, did same experiment, and because these in RAG mice, they don't have adaptive immune cells. They don't have T cells or B cells. And we found we had similar findings suggested that the innate immune cells played a role in exacerbation of exacerbation of inflammation. And we screened the gut immune phenotyping. We tried to understand which immune cells or cell types were affected, and then we found that a cell population called group 3 innate lymphoid cells. So they are immune cells, and they do not express immune cell lineage, such as 11C, 11B, or either T cell, CD4, CD3, something, but they express Cy1, and they highly express rho gamma T and CCR6. So they produce lots of cytokines, IL-22, but they do not produce, in the naive mice, they do not produce interferon gamma or TNF-alpha or IL-17. And these IL-C3 cells were reduced when we treated mice with indomethacin, and this is mediated by EP4, because when we co-treated mice with indomethacin and EP4 agonists, they didn't affect compared to the vehicle-treated mice. So suggested the PGE2 EP4 signaling is important for the homeostasis of IL-C3 cells. We also did another set of experiments to see whether the EP4 is important for IL-22 production secretion. So we treated mice with indomethacin or EP4 antagonist and used the LPS to induce IL-22 secretion. So we found that both indomethacin and EP4 antagonist can nearly completely suppress IL-22 production from LPS-treated mice. As previously reported, that indomethacin can enhance DSS-induced colitis, and these could be suppressed by recombinant IL-22. So we proposed that the PGE2 signaling through EP4 in IL-C3 cells stimulate IL-C3 cells to produce the reparative cytokine IL-22, which maintain the gut wall homeostasis. So this is an example of lymphocytes, and so we showed that the PGE2 signaling in the adaptive T cells had no effect, but in the innate lymphocyte they have protective. And then how PGE2 signaling in myeloid cells affect the gut. So we crossed CD11C mice with EP4 phenolic mice, and then we treat the mice with DSS, and the depletion of EP4 in myeloid cells worsens the gut damage. So suggested that the PGE2 signaling can act on different enteral cell types, such as epithelial cells, myeloid cells, and innate lymphoid cells, and to promote gut epithelial homeostasis, and may also protect against injury. This is one of the story that how PGE2 works as a reparative hormone. But as PGE2 is a very well-known inflammatory mediator, so we wondered whether PGE2 had any harmful effect during gut inflammation. We noted that in the genome-wide association studies, and they found that the PGE2 EP4 is one of the biggest gene that their mutations are associated with Crohn's disease, but less with ulcerative colitis. And the mutation in some alleles is associated with upregulation of EP4 and more severe disease phenotype. we observed that the expression of PGE2 synthase was upregulated in Crohn's disease, and this is associated with the response to anti-inflammatory treatment, such as anti-TNF treatment. So in the responsive patients after treatment with anti-TNF, the COX-2 and the MPGS, they reduced the synthase expression, but in the refractory patients, they didn't change. So they may suggested function of PGE2 in the Crohn-like disease. So we use another kind of model, then we use the T-cell transfer model. So what we did is we isolated naive T cells from wild type or T-cell EP4 knockout mice. So, and then we transferred this naive T cell into the recipient RAG knockout mice. These mice do not have T cells. So when transfer naive T cells, they will go proliferation and differentiation in the pathogenic cells, and then get inflammation in colon and the small intestine in several weeks. So we previously showed, and we have all that the T cell, EP4 deficient T cells, they had kind of prevent the disease, and they associated with reduced Th17 cells, but increased regulatory T cell in the colon. To understand whether PGE2 signaling in myeloid cells had an effect in this model, so we did another experiment that we took naive T cell from wild type mice and transferred to the different recipient RAG2 knockout mice, so the EP4 wild type or EP4 myeloid cell EP4 knockout mice. And in this, so we similarly found that the EP4 deficient in myeloid cells, and they had also prevented the colitis development, in which it also associated with reduced T cell proliferation in the colon, and also reduced enhanced the balance of homeostatic T cells versus inflammatory T cells. So now we asked whether PGE2 had an effect on gut regulatory T cells. We did simply, we treat mice with the COX inhibitor in the medicine, and then, or some mice were co-treated with PGE2 receptor agonist to singly activate the receptor, and then we took the gut tissue or lymph node or spleen and analyzed the regulatory T cells, and we found that especially in the gut, in large or small intestines. So if we blocked endogenous prostaglandin, and they had more FOXP3 positive regulatory T cells, and if we co-treated with especially EP4 agonist, and they have reduced the regulatory T cell in the gut, but they didn't affect the Treg in the lymph node or spleen. Because the gut had lots of bacteria, and these bacteria could affect the toll-like receptors and activate lots of different signalings, especially NF-kappa B through MyD88 and TRIF. So we asked whether microbiome and had functions. So we treated some mice with antibiotics, or we used the MIT88 and TRIF knockout mice, and we found that if we deplete the gut microbiota, or if we block the microbial signaling, and then the PGE2 didn't affect gut regulatory T cells. So suggested that the PGE2 inhibit gut Tregs is mediated by the gut microbiota. So to understand what microbiome will be associated, so we just simply took the gut content and isolated genomic DNA, did 16S sequencing, and these profiling results suggested that the PGE2 is signaling is associated with reduced short-chain fatty acid producing bacteria. So we know that short-chain fatty acid is important for the regulatory T cell development in the gut. And so then we asked whether the PGE2 modified gut microbiome affect the intestinal Treg or gut response. And so we did microbiota transfer experiment, and we treated mice with indomethacin or indomethacin plus EP4 agonist. And then we took the serum content and then transferred to the recipient mice. And these mice, the original microbiome was depleted by antibiotics, and then we induced the DSS colitis. And yeah, the results were very clear that if the mice received indomethacin treated microbiome, and they had very significantly reduced DSS colitis, and if the mice treated with received the microbiome from vehicle or from indomethacin and EP4 agonist treated mice, then they had they normally developed the colitis, and this is associated with change of the regulatory T cell in colon. So then we asked what the PGE2 signaling in which cells modified the gut regulatory T cells. And so similarly, we used the T cell specific EP4 knockout mice, and we analyzed the Treg, and they didn't change between the wild type or knockout mice. So suggested that the other kind of cells, and so we used the menoid specific EP4 deficient mice, and these EP4 deficient mice, so nearly phenocopied the indomethacin COX inhibitor treated, so they had more Treg in the gut. And more interestingly, that 16S sequencing results suggested that most of the enriched microbiome in the EP4 knockout mice, so they are short-chain fatty acid producing bacteria. So it's consistent with our result from indomethacin treated mice. And then we asked how PGE2 function affects the in the myeloid cells. So we treat mice with vehicle or in the medicine, and then we sorted myeloid cells from colon tissue, and we made lots of gene expression and found that the type 1 interferon signaling pathway genes, they are increased by in the medicine treatment. And so we used interferon alpha receptor knockout mice and treated with in the medicine or in the medicine plus EP4 agonist, and that in these mice without type 1 interferon signaling, the PGE2 or EP4 didn't have effects on the regulatory T cells. So we hypothesized, proposed that excuse me, in the normal healthy gut, the microbiome the physiological levels of PGE2, and they have a normal function of myeloid cells. So they maintained a regulatory T cell pool to promote the intestinal homeostasis. However, if they're in the inflamed gut, so they produce excessive PGE2, and then this PGE2 act on EP4 in the myeloid cells, and these produce through so far unidentified mechanisms to regulate the microbiome, and then the microbiome function back on the myeloid cells to restrict their production of type 1 interferons, and then to prevent regulatory T cell proliferation, so driven intestinal inflammation. And if we block the PGE2, if we block PGE2 or block the EP4 receptor in the myeloid cells, and we could have more regulatory T cell in the gut, so maybe promote the resolution of inflammation. So this is so this suggested that PGE2 could act on T cells or myeloid cells, and so far we don't know whether PGE2 has direct effect on microbiota or through affect these immune cells, and to reduce regulatory T cells, but increase pathogenic T cells and leading to development of Crohn's-like inflammation. So take-home message that so we hypothesized that if the inflammation is initiated from the outside of the gut, that means from the lumen side, and then the epithelial damage is the first event, and then PGE2 is likely to be protective. However, if the inflammation is start from the gut inside of gut, that from the laminar propria side, and so disturbance of the immune system at first, and then these immune cells produce inflammatory cytokines, damage the epithelial wall, leading to chronic inflammation. And in this case, we hypothesized that PGE2 is harmful. So I'm going to stop here, and I thank my lab members and all of the collaborators and my funders. Thank you very much. Oh, last.
Thank you, Chengcan, for this excellent talk. Now this data and presentation are open to discussion. I have a general question about this. I was thinking to grapipan, the antagonist which is used in veterinary for osteoarthritis. These for animals, this EP4 antagonist used in veterinary use as therapeutic. In this case, it is similar as yours. That means that there is not only an effect on osteoclast, or it is immune response, Treg increase, which will induce the therapy, inhibit the inflammation.
Sorry, and could you repeat?
I want to, in one of the two last slides, there was a Treg increase, which was inhibiting inflammation due to EP4. So I was wondering if in other pathology, like in arthritis, if there is an antagonist of the EP4 which is used, we don't know exactly, or for also the other inflammatory pathologies that I've shown with aneurysm, with the involvement of EP4, suggesting that the antagonist of the EP4 could be useful. Is it an effect direct on this specific tissue, or it is each time related to Treg role?
I don't think I have the clear question whether EP4 and other tissue, but we did use EP4 antagonist in our the colitis models. Yes, if we use EP4 antagonists, they also inhibit the T cell initiated inflammation.
Mm-hmm. Yeah. Okay. Thank you. There's one question. Yeah.
So very nice paper, Chengcan.
Thank you.
I've two questions. One is in terms of the first example where PGE2 is protective, do you think the microbiome plays a similar role in the NSAID-induced enteropathy that occurs a bit further up the gut? Or have you looked at the immune microbiome interface in just NSAID-induced enteropathy?
Yeah, I think because our microbiome transfer model, we only use the inner medicine treated microbiome, and we are trying to use the 11C mediated EP4 knockout mice to do transfer and to double check whether this microbiome shift is induced by PGE2 or by the off target of COX inhibitor.
Yeah.
Yeah. So now I can't answer your question.
Okay. I mean, it might be interesting to pursue. The other question was just a sort of observation comparing the single cell analysis that you presented in the gut with the single cell analysis that you presented in the cancer cells. And a difference between the two was that in B cells, in Shuh's cancer single cell analysis, EP4 was expressed and EP2 was not. And in your single cell analysis, EP2 was expressed in B cells and EP4 was not. So I just wondered if either of you had thought about what PGE2 is doing in B cells.
This is a very interesting question. this is, to be honest, that's not our data set. We mined from public data sets. And because that immune cells, the gut immune cells, you need intensive digestion. So they may have biased.
Yeah, well, that's what I was
yeah.
wondering.
So I can't 100% sure that the B cell in the gut don't express the EP4. Yeah.
Well, it just appeared to be an interesting contrast.
Yeah. Thank you.
Okay. If there is no more question, it is the end of this symposium. And we would like to thank all the invited speakers for coming and their presentation, and we would like to thank also all the participants of the symposium. Thank you very much.